In this video, we will be learning about the cell cycle. Before we learn what the cell cycle is, let's talk a little bit about how cells know when they need to divide. So a key idea of this unit is that large cells can't receive needed materials fast enough to ensure survival. So you may remember doing a lab like this one shown at the bottom here, your freshman year, where we took this agar, which is kind of like gelatin, with a special indicator called bromothymol blue, and we put them in vinegar. Now, as the vinegar diffuses into these blocks, the blue color changes yellow. We can see that small blocks of agar were able to have complete diffusion, so all the materials were able to travel through those blocks. However, the large cell, you can see, did not have complete diffusion, and only the edges turned. So if this was an actual cell, the nucleus wouldn't receive the materials it needs to survive. So smaller cells receive materials faster. We can also see that here with these cubes. This is one large cube, and it has a surface area that allows for diffusion. But you'll see if that same volume was divided up into many small cubes, we get a much bigger surface area. So there's a whole lot more places for materials to enter the cell. So that's things like food and oxygen can enter the cell, and waste products can leave. So rather than just getting bigger and bigger, the cells are going to divide through a process called mitosis to make identical copies of itself. Now, before a cell can make an identical copy, it must have enough DNA to do so. So before the cell divides, DNA must be replicated to ensure that each new cell has a complete genetic code. We'll be focusing on these ideas of DNA replication and cell division during this whole unit. So what are some of the key roles of mitotic cell division? So that's cell division through mitosis. Well, if you're a unicellular organism like this amoeba here, you use mitosis to reproduce asexually, to essentially divide in two and create a clone of yourself. If you're a multicellular organism like me and you, or this starfish over here, you can use mitosis to either repair and replace. As you can see, that starfish is a leg and it's regrowing the rest of its body. Or you can grow and develop from a fertilized egg to an adult, as you can see down here. So growth, repair, and reproduction are the main jobs of cell division. And remember, mitosis does asexual reproduction. There's a different type of cell division called meiosis, which is used for sexual reproduction, as humans do. So, what exactly are the stages that a cell goes through when it is preparing to divide? Well, that is called the cell cycle. The cell cycle is a series of events that the cell goes through from its creation to its division. So here you can see at this point, we are creating new cells. So this new cell is going to progress through a series of steps. G1, S, G2, and then when it's ready, it will do mitosis and physically divide in two, creating two new daughter cells. So in this video, we're going to be focusing on what happens in that G1, S, G2, and M phases. So let's start off with G1. G1 is the first stage of interphase. Interphase is a group of portions of the cell cycle when the cell is not dividing. So this newly created cell needs to grow and carry out normal metabolism. So when I say normal metabolism, that would be things like cellular respiration. It would need to make proteins, okay? So the basic jobs of the cell to stay alive. And the reason it needs to grow is it was just created from one cell. So it has half the cytoplasm. It needs to increase the amount of cytoplasm, increase its organelles so that it can get to a fully functional size. Now, most cells in our body are actually in this kind of G1 phase because only if a cell receives a signal will it then progress to the next stage of the cell cycle, which is the S phase. If it doesn't, it's kind of stopped in G1. And we have this special name for being arrested in G1, which is called G0. So G0 is an arrested phase. So here you can see the new cell was just created at this point, and we had our tiny new cell, and over the course of G1, which is, I should say, is also called gap 1, but I always remember G1 for growth because that's the main thing that happens. So the newly created cell is going to grow, okay? But if it doesn't receive a signal, rather than going on and going to S phase, it's going to kind of hang out in G0. And it's just going to basically do its normal metabolism to stay alive. But if a signal is received, then it can go and progress to the S phase. So basically, your cells only grow when they need to. Okay? There is a disease where your cells grow when they are not supposed to, and that is called cancer. And we will learn more about that in this unit as well. So G0 is kind of basically stopped doing normal functions. So let's say the cell receives a signal. So let's say it's a cell in your skin of your finger and you get a paper cut. The neighboring cells that are damaged from the paper cut will release signals called growth factors, and they'll tell their neighbors, hey, you need to divide so you can make new cells to replace the damaged ones and to fill in the cut. And so if this cell in your finger receives a signal, it'll leave G0 and it will enter the S phase. The S phase is the second stage of interphase. Okay? It is when the cell replicates its DNA, and it does that so there will be a copy of each of the chromosomes, each piece of DNA, so that there is enough DNA at the end of mitosis for each of the two cells. Sorry about that. I caught the typo in that part. It says cell replicates its DNA, so there will be a copy of DNA for each of the two daughter cells created at the end of mitosis. It's called the S phase because the job is to synthesize more DNA. So let's look at this picture and help us understand that a little bit more. This cell right here has 2 chromosomes, okay? And on that chromosome is gene A. So you can see the blue chromosome has a dominant form of that gene, and the red chromosome has the recessive form. Now, before DNA replications, these are just single strands of DNA. After DNA replication, we have something that's created that are called sister chromatids. A sister chromatid is an identical strand of DNA made through DNA replication. And they are often joined at the center called the centromere. Okay, so sister chromatids are at the centromere. So you can see they both have this dominant form of gene A, which means they're genetically identical, and all the other genes on that chromosome would also have the dominant form of the allele. For our kind of purpley red chromosome, we also have sister chromatids, and you can see they're also genetically identical to each other and attached at the centromere. So we still have 2 chromosomes. They're just replicated chromosomes. Okay, so they are sister chromatids attached at the centromere. Now those sister chromatids will be divided up so that there's 1 blue and 1 purpley red colored chromosome for each of the daughter cells made. So just to recap, S phase synthesizes more DNA. It creates sister chromatids. So all those S words will help you remember their job. In G2 phase is also called gap 2, or it's another stage where there's growth. It is the 3rd stage of interphase. And at this point, the cell is going to make sure it's the right size and it has all of the materials necessary to divide. Okay, so we have one cell that's going to divide into two cells, like you can see here, and each of those two cells are going to need resources in order to survive. So that means during G2, when it's preparing for division, it's going to be making new organelles, it's going to be making more proteins. Whatever those two new cells need to survive, they're going to get made in G2 so that they're ready to go. Now, once the cell is all ready, it's copied its DNA, it's grown to the right size, it's made enough proteins and organelles, it can then progress on to the M phase. The M phase gets its name because it is when mitosis occurs. So mitosis is nuclear division. So that is when the DNA in the nucleus divides. It also includes cytokinesis, which is when the cytoplasm divides. So here you can see mitosis includes prophase, metaphase, anaphase, and telophase. This prometaphase is basically, we'll go into it more later on, but prometaphase is basically just late prophase. Some books call it that, some don't. Okay, so we have four basic parts of mitosis. I call them PMATs: prophase, metaphase, anaphase, and telophase. And that is mitosis. That is explaining how the DNA in the nucleus divides and how you split up those sister chromatids so that one of each sister chromatid ends up in each cell. Now the cytokinesis part is what you can see here when the cell actually divides, and it occurs at the same time as telophase. So what exactly gets created at the end of the cell cycle, at the end of mitosis? You will get two genetically identical daughter cells. So let's just run through this one more time to make sure everybody understands. So we will start with G1. In this case, we have 4 unreplicated chromosomes. So 4 chromosomes, normally in interphase. Okay, the DNA in the nucleus is actually very loose and wiggly, and the name for this kind of loose DNA is called chromatin. But in this picture, they condense the chromosomes to help you understand that there are 4 chromosomes. So we're starting with 4 chromosomes. Okay, the S phase will make sister chromatids because it's going to synthesize DNA. And here now you can see there's 4 replicated chromosomes. Although those are stuck together, those chromosomes look like this. So we have 1 sister chromatid and a second sister chromatid. Now we still have 4 chromosomes. They're just replicated chromosomes at this point. Now in the M phase, what's going to happen is you can see that they're more condensed. You can definitely see that they're more X-shaped in this M phase at the start of mitosis. Those replicated chromosomes are going to divide. So what's going to happen is those X-shaped chromosomes are going to get pulled apart. So one sister chromatid will go to one cell and the other sister chromatid will go to the other. So you can really see here, although there are 4 chromosomes, there are 8 chromatids, sister chromatids, so that they can be separated into 2 cells. 8 chromatids divided into 2 cells means there's 4 chromosomes each. Now, these daughter cells that are created are genetically identical to what we started off with. So not only do they both have 4 chromosomes like our original parent cell, but the sequence of DNA in those chromosomes should also be genetically identical. And what that ensures is that as the body is doing mitosis and it is growing or repairing, every new cell in the body that is created is going to have all the chromosomes it needs, a full blueprint for making you, so that each cell has the information it needs to do its jobs.